So I never know why people applaud before I talk, but thank you very much. So I'm told that at 9:30 the fire alarm will go off. So that has, I don't think that has it's a commentary on what I'm saying, or it might be, but by then. So consider a country which I had to point out to Munir, it does include the Shetland Islands. So his map didn't include that. So the reason that we have large biobanks in precision medicine is what I call the paradox and the opportunity. And that is, if we're going to treat patients differently from average, we have to develop evidence for that. And if we're going to develop evidence, then we have to have a large denominator from which to draw small groups of patients who look different from average. And those kinds of small groups could be pharmacogenetic outliers. They could be patients with unrecognized Mendelian disease. I'll talk a little bit about both of those. We like to say in our biobank that we can't find patients who have only one disease, and we also can't find patients who take only one drug. I think out in the practice world that may not be completely true, but certainly in a tertiary care hospital, that is the way things work. And then even if we're taking care of common diseases like diabetes or cancer or atrial fibrillation, we recognize that there are subsets within those large populations of patients who might benefit from treatment different from average. So we're very interested in finding those subsets and understanding them better. So this is my United States map of that vision, and this is where Tennessee is, where I live now, and Nashville is the little star. And so in Nashville, what we did 15 years ago is we started to create this resource. And the resource started with taking our electronic health record, which is around 3 million records, and de-identifying it. And de-identification is a word that requires another hour or two of discussion. I'm not going to do that. And then a subset of those samples have DNA samples attached to them. And it's around 250,000 now. I would love to say that they've all had whole genome sequencing. That's only possible in the United Kingdom. It doesn't happen in our country. So the things that we have learned is that we can find cases and controls using electronic algorithms. That was not self-evident at the beginning, and it is. We can export those definitions across electronic health records, and we know that because we work in something called the eMERGE network, the Electronic Medical Records and Genomics Network. We've been doing that for about 10 or 15 years, now 12 years now. And so what we understand is that an algorithm that we find to find patients with diabetes or to find patients with whatever you want, we develop it at our place. We can export it to other electronic health records, and it works. We can replicate known signals for disease. That was an early study, and that was really important to show that the electronic health record is, in fact, a usable resource for genomic medicine. There were people who were skeptical about that. And then we can study variable drug responses. Of course, Munir's already alluded to the fact that we spent a lot of time doing pharmacogenetics and pharmacogenomics in my environment, and I've spent most of my career doing that. So we have spent time looking at using these kinds of resources to study pharmacogenomics. I'll just point this one out here because I'll come back to that sort of roughly at the end. And this is a more recent version of that. And I think the important thing about pharmacogenetics within an environment like this is that it's much more complicated than disease genetics. So I can find patients with disease X at a point in time. What I have trouble doing is finding people who have disease X, drug exposure Y, some kind of drug response assessed in the electronic health record prior to the drug and then on the drug. And so we've spent a lot of time trying to figure out the time dimension of this story that many patients have. So this is a more recent example. The numbers, you could say the numbers are large or the numbers are small. So these are hundreds of thousands of patients, and out of hundreds of thousands of patients, we can find 3,000 who have an acute coronary syndrome event while they're taking statins, and a set of matched 7,500 controls who don't have an event while taking statins. We do a genome-wide association study. Clear signal at the Lp(a) locus. Lp(a) is a target for hyperlipidemia now. So that was sort of very gratifying, and one of the many pieces of evidence now that Lp(a) is an important target in cardiovascular medicine. We also discovered that once we've genotyped somebody for some project, the genotype data are there and they can be used over and over again. It seems self-evident now, but it wasn't self-evident at the beginning, I can tell you. So this is one of our first studies. We were studying electrocardiographic intervals, and we reused a lot of data from the eMERGE network that had been genotyped for the patients had been genotyped for other indications. And I point out this word phenome-wide because that was one of the big advances that we made along the way. So in the course of an ordinary genetic association study, what one does is one identifies a trait, a disease, a drug, a complication of drug therapy, whatever, and then uses family structure or whole genome sequencing or exome sequencing or whatever you want to understand the associated genetic loci, the associated genes, associated variants. So we realized as we were collecting sets of data of large numbers of patients with electronic health records and dense genomic information of this type, that we could turn the experiment on its head. And we could say, let us ask the question, with what human phenotype is a particular genetic variant associated? So that was the first idea. And I can talk about this for a couple of hours, again, choose not to. But we spent time in the eMERGE network validating this idea. And the signals that we get by genome-wide association are replicated by phenome-wide association. So this is actually a very cool study. This is a variant in IRF4 that was associated with hair and eye color. And so we looked, using phenome-wide association, at what phenotypes were associated with that. Actinic keratosis was a very, very strong signal with a p-value of 10 to the minus 27th or so. Lots of non-melanoma skin cancers over here. And then this one here, solar dermatitis. And so I'm fond of saying when I show this slide that solar dermatitis is the way sunburn is coded in the electronic health record. So this is the slide that tells you that people with red hair sunburn more easily. And so it's a complicated way of getting at that phenotype. So more recently, the term phenome-wide association, PheWAS, has been adopted by the genomics community. And so we're not the only ones who do this anymore. This is a paper that was in late 2018. And what they did was they did an experiment that many of us have done. They took variants in genes that encode drug targets, and then ask the question, number one, if I do a PheWAS, do I retrieve the disease for which the drug is indicated? And then number two, do I retrieve any other diseases which might be an opportunity for drug repurposing or drug repositioning, or might identify adverse drug events that you might see? So this is the known phenotype. This is the known disease for these genes that are the targets. And these are the new phenotypes. And the reason I show this slide is to show that this is one use of phenome-wide association, and the other is to show you the numbers. So this is UK Biobank data, 23andMe data, a couple of other large biobanks, and the numbers that you see now are not 10,000 or 15,000. They're hundreds of thousands, approaching millions. So when you have these kinds of large data sets, you can start to extract signals. You can also ask whether the signals that have a p-value, an odds ratio of 1.05 are useful or not. But it turns out that in the drug industry, they are very interested in developing evidence that human genetics supports a new drug indication. So this is a really important part of drug development now. So this is the idea of the phenome-wide association study. And we realized after a while that the question we were asking was limiting. We don't have to actually, once we have this, we don't have to ask this as the input function. We can use lots of other input functions. We can use sets of genetic variants together. We can say with what disease, with what other diseases does this disease or this drug exposure associate? So that's the curated EHR phenome is the key. So I'm going to show you an example from here, a couple examples from here. So when I'm not worried about personalized medicine, I worry about this particular adverse drug event. I've studied this for a couple of decades, more than a couple of decades, the idea of a drug-induced long QT syndrome and torsade de pointe tachycardia. We have developed a system at our place that as soon as a house officer writes a note and they use the word torsade, they push save, and I get an email. And the email says, the word torsade is in the progress note. Go back, and this is the medical record number. This happens to be the medical record number of this patient. So this is a way we have of actually finding cases in our hospital. And it turns out I thought I knew about all these cases, and of course I don't, because it's a big place, and there are probably 4 or 5 times more cases of drug-induced torsade at our hospital than I ever realized. So as I said, I've studied this for a couple of decades. Some patients have unrecognized congenital long QT syndrome, another disease I'd love to talk about forever. We have a common variant, 1 to 2% minor allele frequency, that does seem to increase risk. There's a reason for that, we think. But otherwise, we really haven't found single variants that confer high risk for this adverse drug event. So that's where the story sort of sat until about a year and a half ago, when we started to look at genome-wide association data. So this is the QT interval in normal people in our electronic health record. So there's a distribution from about 340 to about 470. What you can do with those kinds of data is you can ask the question, what is the genetic or genomic basis of variability in that phenotype? So there are many, many loci that you can discover when you do a genome-wide association, like a study like this with large numbers of patients. The top locus is variants near a gene called NOS1AP. The physiology is not well understood. We have ideas about that now, but that's another story again. So the p-value is 10 to the minus 213th. It's actually not the lowest p-value I'm going to show you today, but pretty low. But the effect size is tiny, 3.5 milliseconds per allele. So remember, when you look at an electrocardiogram, you're looking at you used to look at a pen. Of course, you look at an electronic image of a pen, drawing a line, and the pen width is less than 3.5, around 3.5 milliseconds. So you can't see this. A human being can't see this. So this introduces the idea of genetic risk scores. So you take all the variants, and people argue about how many is all. It could be the top 60, it could be the top 6 million, and you make a genetic risk score. So the score for an individual subject, I was a math guy in college, so I have to show this, but the score for an individual subject is all the variants, and then for each variant there's a term that says, you have 0 risk variants, 1 risk variant, or 2 risk variants, and then an effect size. So the effect size for NOS1AP would be 3.5 milliseconds. So you add all that up and you get a genetic risk score. And so the idea is that genetic risk scores will separate people at high risk for something versus people at low risk, or cases versus controls, or whatever you want. And here's sort of a little cartoon that shows that people at this end have more high-risk alleles and people at this end have more low-risk alleles. So we actually applied this to a set of patients we had collected with drug-induced torsade de pointe, and we expect a p-value of 0.05 to separate the cases and controls, and we got a p-value of 10 to the minus 7th, and they separate quite nicely, the cases and the controls. So there's some utility in using this, we think, to find people who are at this end of a genetic risk score distribution. This has become a very popular notion based in part on this paper that appeared late last year in Nature Genetics. So Say Katharison and friends mined the UK Biobank, and what they did was they looked at genetic risk scores for coronary artery disease, and so you can make a distribution. You can find people who are in the top 0.5% of the distribution, and their risk of coronary disease is the same as the risk of somebody with a monogenic familial hypercholesterolemia mutation. And then they did that for atrial fibrillation, type 2 diabetes, inflammatory bowel disease, and breast cancer. And again, there's always this set of people up here. So the question of sort of what do you do with these data is actually an open one right now. But at one of our lab meetings a couple of weeks ago, one of my junior colleagues who has been working on this problem sort of said, he presented 2 scenarios. He said, here's a 50-year-old man who goes to the cardiologist, says, I want to know what I need to do to stay healthy. I brought my 23andMe genotyping data with me. So cardiologist number 1 says, let's check your lipids, stick you on the treadmill, see you next week. A week later, you have a 12% risk of coronary disease in the next 10 years. You need to exercise and start taking a statin. Cardiologist number 2, much more with it, the Twitter guy. 5 minutes later, he sticks in the coronary artery disease risk profile, and he says, based on your genetic risk, you have a 12% incidence of coronary disease. You need to exercise and start taking a statin. So this is facetious, and we don't know quite what to do with these data, because remember, this one you could do when somebody's 20 years old. This one you can't. So by the time somebody has all these risk factors, the genetics may not be all that important, or the genetics may have already played out. But early in life, the genetics are going to predominate. So we think that we're going to have to find our way where this plugs in. I'm mindful of the time, and I'm mindful of the fire alarm. So Jonathan Moseley, who was the one who actually was responsible for the slide I just showed you, has this other idea, which is a little complicated, but I'll try to run through it quickly. So if you're interested in studying a biomarker in the electronic health record, what you would do is you'd look in your electronic health record, find people who had the biomarker measured, and then associate the biomarker concentrations with some set of diseases, and then you have developed a biomarker and you have to validate it and deploy it in practice. Sometimes, though, the biomarker that you're interested in has not been measured in the electronic health record. So here's the idea. You take a group of healthy subjects who have had your biomarker of interest measured, you measure it. Those people have had genotype data done as well, so you can develop what we call a multi-SNP predictor that tells you what the biomarker would be. Then you deploy it in an electronic health record with tons of genotypes, and you get what we call a virtual biomarker, and then you can do this association. So does that work? Well, we have at least one example where we think it does. So this is the idea. You take these data from Framingham or some large data set like this, the UK Biobank, for example, and then you deploy it in a population like BioVU or eMERGE. So what we did was we looked at the biomarker TSH. TSH is measured in patients who have known or suspected thyroid disease, but it's not measured across the population in everybody. But it's been done in Framingham and other places. So we developed a multi-SNP predictor, deployed in the electronic health record, and this is the way that these data are displayed. The colored symbols are statistically significant, and this is a so-called volcano plot. And what you see is that low TSH associates with hyperthyroidism, high TSH with hypothyroidism, and the only disease that pops up is atrial fibrillation. And this is one of 3 papers that now shows an association with variability in TSH in the normal range and susceptibility to atrial fibrillation. So it's an interesting way of using the electronic health record, which I must tell you, when we started 10 years ago, I wouldn't have dreamed that we would do that. So I want to say something about phenotype risk scores. So that's something that we've popularized at our place, and I'm very, very excited by, because it gives us the opportunity of using the phenotypes in the electronic record to find people who will benefit from genotype approaches. So here are 3 patients, and we're looking at the disease cystic fibrosis. So here's a patient with lots of symptoms that are suggestive of cystic fibrosis. Here's a patient with none, and here's a patient with a couple of symptoms. So what we do is we create a phenotype risk score, not complicated. Do you have the phenotype or not? You don't have it or you have it. And then there's an effect size. Now, what is the effect size? So for bronchiectasis, pretty specific. We give it a large weight. Asthma, less specific, so we don't give it much of a weight. And then we do this sigma thing, which is which is just adding up all the symbols. So you add up all these scores and you get 8.2. You add up all these, you get 3.4. And then you can do a normalized risk score. And here are we deployed that in our electronic record in a group of people who we knew have cystic fibrosis or don't. The controls have very low scores. The cases have very high scores, although there's a distribution. And we did that in 6 diseases. So this is the lowest p-value I'm going to show you. So cystic fibrosis, the p-value was astoundingly good. The one disease that didn't work in was phenylketonuria, until we realized that nobody has symptoms from phenylketonuria because they get diagnosed at birth, they get put on the diet, and they don't get the symptoms. So this is like our positive control. We're very proud of this, although that's not the way we planned it in the first place. Here's an example of a particular disease that I'd never heard of called nephrotic syndrome, type 7 nephrotic syndrome. This is a stop codon in a gene called DGKE. And here are a group of patients. Here are the symptoms. Here's the weights. And here are the patients, male 24 years old, male 36 years old, what have you. And these are the individual risk scores. So some people have no symptoms and a score of 0. Other patients have a very high score. This one here has a very high score. And in fact, this one here, this is a recessive disease. This patient had a very high score and, in fact, has 2 variants. But what's interesting is that there are these patients here who have one variant and nevertheless have higher scores than average. So we think that they either are digenic, they have an undetected variant on the other allele, or they have another gene that's playing into this. But it gives us an opportunity to identify patients who look like they have genetic disease and would benefit from a closer look. So we looked at a set of 21,000 people who had had dense genetic data done, and we found 18 associations between a disease and 18 examples of patients who have high phenotype risk scores and a variant in the right gene. Those 18 examples, most of them were multiple patients, so there were actually 800 patients out of the 21,000, so it's about 3 or 4% of patients look like they have a genetic disease, and that was unrecognized. One of the questions was biologic validation. Did they have functional data that showed that these variants actually alter gene function? In the original paper, we had some data on that. We have more data on that now. And then I want to just tell you quickly about a story that's going on in the eMERGE network. So this is a cartoon that I'm fond of showing. Whenever I talk about personalized medicine, anybody can adapt it if they want. So in the eMERGE network right now, the project is sequence 109 of what I call your least favorite genes: cancer susceptibility, arrhythmia susceptibility, cardiomyopathy susceptibility. And then we return likely or known pathogenic. That's a tiny proportion of all the variants we find to patients and physicians. And then we'll use the electronic record in other ways to help figure out what the variants of uncertain significance whether they associate with a phenotype. So at Vanderbilt, we just have our data now. At Vanderbilt, 6.8% have a returnable variant. And it turns out 2/3 of those are cancer susceptibility. And we have wonderful stories of people who are detected as having cancer through this mechanism. And I'm sure that as the genetic approaches in England or in the UK expand, you'll have equally compelling stories. So one of the things that we've now started to do is look at variants of uncertain significance. So FBN1 is the disease gene for Marfan syndrome. 25,000 people have been sequenced, and we have about 95 or 97 variants in FBN1. 5 of them are known to be pathogenic, and they carry high risk scores for Marfan syndrome. And 7 or 8 are known, 9 actually, are known to be, but are thought to be benign, and they carry low risk scores for Marfan syndrome. And now we have 82 others that are variants of uncertain significance, unannotated, and here are the risk scores. So there are 2 that we think are pretty clearly pathogenic, and there's a bunch that are probably not pathogenic, and then there's a bunch in the middle. So we think the electronic record can be used as a tool for functional genomics to figure out whether somebody has a rare variant, but you only get this if you share data. If you have large data sets, and you can start to share data. You can't do it on your own. So a little bit about implementing pharmacogenomics at the end. Again, I can talk about this for a couple of hours, and I won't. But here's a rant before I start. So again, I'm a long QT guy. So this is an 82-year-old man who is on sotalol for paroxysmal atrial fibrillation, develops renal failure, and because of renal failure, the drug is not excreted, plasma concentrations go up, and he gets torsades de pointe. And if you look at the label for sotalol in the FDA label, it says because sotalol is excreted predominantly in urine, it's terminal elimination, blah, 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 you need to adjust the dose. And every clinician knows to do that. And every clinician knows that if they don't do that, they'll get into trouble with the patient and perhaps with the lawyers, at least in our country. But there are no randomized, and no one's ever going to run a randomized trial to do this. Of course, in the genetics world, you have to run the randomized trial. So fast forward a decade, we now have a program where we preemptively genotype people for key pharmacogenetic variants. We implant them in our electronic record. This is a screenshot of our EPIC electronic record, which is new and we're still getting used to. And we have developed clinical decision support so that when somebody is prescribed clopidogrel and they have a CYP2C19 variant that predicts that they will not respond as well as they should, then the physician or the provider is given the option of ordering alternate therapies. So in our program, when we looked at the first 13,000 patients who had been genotyped, there were 2% who were homozygous for loss of functional alleles in a gene called CYP2C19, which is the key gene that activates clopidogrel. So these patients are predicted to be giving them clopidogrel like giving them placebo. These patients are heterozygous for loss of functional alleles, so they have less of a response, and these are people with no variant. These are the 5 variants that we looked at, because these are the common ones. Common in European populations, common in Asian populations, this one here. So, but what I want to tell you is that if you look in large databases, this is one of the large databases we use, there are hundreds of non-synonymous variants in CYP2C19. Some of those must be loss of function, and some of those must account for some of the variability in drug response that we see, but we don't have a way yet of getting at function. So that's a challenge for the field. When we looked at 5 drug gene pairs, not just clopidogrel and CYP2C19, but others, we found that 91% of patients have something that a provider might want to know when they were prescribing a particular drug. So that's the argument that says everybody has something. You'd like to know about it before you get the drug, and that's why we think preemptive pharmacogenetics is going to be the way to go. Everybody wants to have the other problem is with genetics, and especially in pharmacogenetics, is that physicians and others want to be told, this patient will respond to this drug, this patient will not. This patient will die from this drug, this patient will do well. And the fact is that drug responses and other human phenotypes are along a continuum. And the genetics tell you whether you're going to be this kind of patient or that kind of patient. They shade the odds. They don't actually tell you whether somebody's definitely going to have one outcome or another. So I'm fond of showing this slide. This is a non-electronic medical records room at Vanderbilt probably 20 years ago now. And what I hope I've shown you is that using the electronic health record and using tools of modern genomics, we can do both discovery and we can start to implement those discoveries for patient care. And I won't read them to you because I've read all that. The last time I was in England, I had been invited by Munir to go to a meeting in Liverpool, and this was the headline in the newspaper that was in my room. So I'm jealous, to make a long story short. You have made a big investment in genomics. I think it's a journey. You're still trying to figure out exactly where things will fit in. And I think that as these very large data sets are developed, it's important to get really smart people to think about how to use them in unusual ways. The phenotype risk score is an idea that we would have never thought of when we started, but here we are, and I think that that's an opportunity both for us in the United States as well as you and anyone else around the world who's developing these very large data sets. This is the team. It's a team sport. Let me just highlight two people, Josh Denny, who's my partner in crime for the last decade and who is now the principal investigator for the Research and Data Core for our answer to Genomics England, and that's the All of Us initiative that's recruiting a million patients across the country. And Lisa Bastarash, who is the person who invented the phenotype risk score, and Jonathan Mosley, who's the crazy man who has the cardiologist, whose 50-year-old patient goes to the cardiologist. Jonathan turns 50 next year, so maybe that's why he took that example. So thank you very much for the opportunity to tell you a little bit.